I'll start by thanking the real winners. So thank you, BSCB, for this award. I think I'm quite humbled, quite privileged. But also, I think the real winners are these people. They are my team. They keep the show up. A developmental cell biologist lab, and we work on this organ heart. So our work kinds of span kind of across scale. So we work at cellular scale, tissue scale, organ scale, and also extracellular matrix and everything which is kind of outside the cell. So for today's talk, I'm going to kind of, because these systems have been extremely powerful. You can visualize, you can manipulate, you can poke, squeeze, whatever you want to do. But we also know that not all tissues and organs are simple like this. And actually, they have quite complex anatomy. For example, things of branches, tubes, and ridges. And they also have quite complex physiology. And they undergo extreme mechanical deformation as part of their normal physiology. Think of your breathing lungs. You're breathing all the time, and imagine the cells or tissues inside your lung, what they're kind of experiencing every time you breathe in or breathe out. Think of your heartbeat, and all of us here, I think the heart is beating at different rates. Of course, mine is beating at a much higher rate, but still, like it's constantly undergoing that kind of remodeling while the heart is beating, or your blood vessels, or your gut. Um, so yeah, there is quite remarkable deformation, but yet these processes they happen at the right time, right place. These structures are built to the right proportion and to the right size. So, how does morphogenesis cope with such extreme chaotic dynamics and still build robust structure? That is something which is quite interesting and keeps us kind of focused on this problem. So you might have guessed, or by that time you know now, that we use heart as a model system to understand this problem. Of course, heart is an extreme example. It beats around 100,000 times per day, kind of pumps all that blood throughout your body. And of course, there's a lot going on here, like the heart stretching, compressing, shear stress, you name it, everything is kind of like going on here. Um, but for the heart to kind of acquire its complex form and function, it has to undergo all those processes like shape changes, weight specification, and all that while the heart is beating. And this process has to be robust because it's crucial for life, so mistakes cannot happen. And that's why this makes this problem of morphogenesis here quite challenging, but at the same time quite extraordinary. And we want to decode that problem here. We use zebrafish as a model system. I'm sure I don't need to convince you why zebrafish, but actually we can visualize things as they happen. It's extremely tractable genetically, but also biophysically, and also in the case of heart, one cool thing is that zebrafish can survive without a functional heart for up to 1 week of its for post-fertilization. So what we can do quite extreme, sometimes crazy manipulations, and we can still have a plate full of embryos kind of swimming around without a beating heart. So we can it gives us a lot of flexibility in our experimental design. Of course, I didn't play the movie, but that's okay. So this is how a zebrafish heart appears at 5 days post-fertilization. So very briefly, it's a two-chambered heart. This is ventricle, this is atrium, and these are the blood cells being pumped by these two chambers. There's a lot going on here, but let's try to focus on inside this box. So what you see inside these structures are these block-like structures protruding inside the lumen of the heart. These structures are called trabeculae, and we focus on them. It's not unique to fish. We also have trabeculae in our hearts. Actually, trabeculae are present in all vertebrate hearts. So what did you do? They are the force generator component of your heart. They make your heart an efficient pump. And if there is any aberration during this process, it has lethal consequences. So in the case of model organisms like mice, fish, or chick, I think they kind of die very early. And in the human, it can lead to cardiomyopathies and various kind of defects. So they are crucial for heart function. Now, for the heart to kind of acquire its complex structure, the beginnings are quite simple, like what happens in development. So in the beginning, it's just a simple tube. It's a linear tube. The tube kind of twists and then rotates, and then it forms these two chambers. And then eventually this process of trabeculation happens. Remarkably, all this is happening while the heart is beating. So heart starts beating as early as 1 day post-fertilization. And I think I'm trying to bring this up again and again for you to appreciate that imagine if a cell has to divide or if a cell has to delaminate, it has to do under these circumstances. So I think that's just quite extraordinary kind of feat. So yes, trabeculae. So very briefly to tell you how this process of trabeculation happens in fish. So, of course, I have simplified the system here. So, I'm just going to show you cardiomyocytes, which are the major muscular cells of the heart, but there are other components here. So, in the beginning, there's just a single layer of cells, as you can see here. Eventually, a subset of cells, they delaminate as single cells to seek the inner trabecular layer. So, basically, from one layer, you end up having two distinct layers, the outer compact, which you can see in gray, and the inner trabecular layer, which is in the red. These single cells, they go on to form these multicellular ridges, and then remodeling happens when these ridges, they kind of fuse and they undergo some sort of morphogenetic rearrangements, which we have no idea, and they form this very complex 3D meshwork. So it's quite a remarkable transformation, and anyone who is interested in shape or morphogenesis, I think it's a perfect playground because you start with a hollow human, and then you fill this human with this complex meshwork. And we try to understand what we're trying to understand how this transition happens. So for today's talk, I'm going to focus on both these layers, the outer compact layer and the inner trabecular layer. And the question is about how they pattern and how they kind of keep in shape. So to solve this problem, in my lab, we kind of ask questions around these themes broadly. So, for example, we try to understand how geometry of the organ itself control morphogenesis, how these complex 3D topology emerge. Of course, we want to understand the feedback between signaling and patterning. How do you make a mechanically resilient tissue? And of course, the nuclei, because the nuclei is kind of sustaining all those forces. So how does the nuclei kind of remain intact? But for today, I hope, I'm kind of giving you a glimpse of these two themes here, and I'm going to kind of very briefly tell you two, I won't call them stories because some of them are in kind of like in flux, but yeah, a couple of stories around these two themes. I'm going to keep it very broad and conceptual level, but if anyone's interested in the details, I'm very happy to kind of talk. Okay, so let's start with geometry. So when I started working on this problem many years ago, when I was in Dinius lab as a postdoc, the first question that was confronting us was that how do you end up having these two layers? Like why cells they delaminate, why they become trabecular layer? Like what's the mechanism? So this is published, so I'm going to keep it extremely brief, but basically we found this is based on differences in the mechanical properties of the cell. I should say this here because somebody, so I come from Alpha Lab. I did my PhD at Alpha Lab. I think people who work on epithelial mechanics, they might know. So when I joined BBS Lab, I had all these alpha influence on me about, ectomers and cell-shaped mechanics. So I looked at this and I was like, this has to do something with the invention of these cells. So I started with like kind of those approaches, and I think in the end we found that indeed this was true. So basically, these cells they're doing classical apical constriction. So they have high ectomass intention on the apical side, and then they delaminate, and that's why you end up having this another layer of cells. So that was quite satisfying. But then what we consistently observed that this process of delamination happens in this particular region of the heart, which is called outer curvature. So basically the first delamination cells, they appear there. And that was kind of like, but why it happens is not known. And that's kind of the first project I started when I started my lab in Crick in 2021. So why kind of delamination is patterned in the outer curvature or that particular region of the heart? So one of the prevailing hypotheses in the field was that there are these particular signaling pathways which are enriched in the outer curvature, and that's why we have delamination there. For the sake of time, I'm not going to show you any data, but we kind of rule this out. So those signals are not there in the right time, right place. And of course, so we thought, okay, what it might be. So we focused on ECM. It's called CECM or cardiac extracellular matrix. It's a specialized matrix primarily composed of hyaluronic acid, which is a sugar. And here we're visualizing this matrix in live using this biosensor which binds this hyaluron. So you can see this matrix inside here. And what we observe that as early as 55 hours post-fertilization, which is when the delamination starts to happen, you see these thin and thick regions. So the asterisks, they mark kind of thin regions. And wherever these thin regions are, you see cells delaminating next to it. So wherever you see a thin region, cells next to it will be delaminating. So that was interesting. We call these thin areas fractures. It's quite a like mechanical fractures, and it will become clear about problem fractures. But we know that they can satisfy all the criteria of a canonical mechanical fracture. We can see them in life. So here you are looking at a beating heart, and you can see these fractures. They're kind of running across the myocardial tissue, and this is of course you are visualizing the ECM. So you can see these fractures live in ECM. So here again, just to help you visualize, you can see these fractures by the loss of fluorescence intensity in here, and we have pseudo-colored the delaminating cells. So you can see the cells literally crawling into these fractures. We also know that if we locally, the ECM, so basically if we locally kind of abrogate these fractures, delamination does not happen. So these fractures are required for delamination. But then the question, of course, I've kind of skipped quite a few things here, but I think I want to come to this part. The question was like, why these fractures happen? What leads to this fracturing? So this is a lot of work actually done by Chris, which I'm summarizing in 3 sentences here. But basically, the first question can be that maybe there are some enzymes which are locally remodeling the ACM. That's not true. Those enzymes are not there at the right time, right place. And if you manipulate those enzymes, the fractures are still there. So enzymatic remodeling is not needed. Other thing can be that as the cells they go inside, they locally remodel the ACM. That's also not true because fractures happen way before delamination starts. And of course, if you block delamination, we still see fracturing. So delamination is not required. So then we thought if you really want to understand how fractures emerge, we needed a detailed understanding of the spatial hemodynamics of ACM. So what Chris did, he came up with this method, which allows us to kind of visualize beating hearts as early as 1 dpf, which is like when the heart is still a linear tube, and that hasn't been done before. It's very difficult to visualize ventricle because the heart is just under the head of the fish. So this is a body expand method which Chris has developed. And what it allows us is that we can actually see fracturing as it happens, and we can go early and see when does the fracturing start. So in the beginning, what you see that the ECM appears quite smooth. And just 6 hours later, you can start seeing these cracks, small cracks here, appearing. And these cracks, they ultimately kind of propagate as fractures. Now, what is happening between these 6 hours? I think you can guess the heart is beating all the time. And we thought that these mechanical forces, which are kind of generated by heart, that can generate fractures. So we quantified 2 parameters. First of all, the heart rate, how fast the heart is beating. And of course, percentage stretch, basically how much the heart is deforming. And both these parameters, they go up between these 2 stages. So that tells us first that this fracturing happens over time. It's not like the ACM is fractured from the very beginning. And of course, there might be a role of heart contractility here. So if that is true, if you block contractility, you should stop fracturing, and indeed it happens. So here we have used 2 independent genetic manipulations, and you can see that in both these manipulations, if you see a controlled heart, you see these thick and thin region. But in these hearts, which have never undergone any sort of beating, you can see that the ECM is much more smoother. And this we also observe in Crick hearts. So we can confirm that, and we could see that the fractures are present in Crick hearts, and if we block contraction, then the ECM stays smooth. So that is quite satisfying. But then, again, because I think we like to think of these processes in a much more holistic manner. So we not only forces are required, but then how does this influence the efficiency of fracturing, the dynamics, and the magnitude and everything? So we teamed up with Alejandro at ML Barcelona, who is a physicist. And basically, we developed a biophysical model. Very briefly here, we are kind of modeling the ECM as a new open material, where you have the limit of strain softening, and that determines when and how fractures will emerge. So what we saw that even in our system, these fractures, the ECM starts fracturing, but most importantly, or rather interestingly, what we see that these black spots, which is fractures, they kind of appear in this outer curvature of the heart. So they start somehow here. So we thought, okay, what might be the reason there? So we thought that this might be because of geometry. So because heart is an anisotropic, it has an anisotropic shape, in shape. And this part of the heart, which is outer curvature, perhaps is undergoing maximum stretch, and that's why you have fracturing appearing there. So how do we improve this? So we calculated strain maps by tracking nuclei and beating hearts. And indeed we see, so this is a heat map, that you have much more higher strain in the outer curvature, as you can see in warmer colors, in yellow color. And because there's higher strain, that's why the fractures appear there, and that's why the lamination happens there. So strain, fractures, and delamination. So geometry is kind of required to pattern mechanical stretch, which ultimately leads to patterning of fractures. But if that is true, you expect that if you take away the information of geometry, so if you kind of linearize these parts, the fractures will change, so basically you will change the pattern of strain or fractures. And indeed, this is true. So if you create an incident, looks like I don't have the slide here, I'm sorry, but I'm just going to tell. So if you create an in silico heart, which is linear, you will see that the strain is changed and the fractures are linear, and that tells you that ultimately geometry is also important there. So kind of summarizing it here, so what we see that this ECM, which is largely composed of hyaluronic acid, is a viscoelastic material. It is layered on top of a contractile myocardium, and because of that, in the beginning, when the myocardium is not beating, you have a smooth layer of ECM. But as soon as it starts to beat, you start seeing these cracks, and these cracks, they become fractures. Because this part of the heart undergoes maximum stretching, strain is highest here, fractures are here, and that's why you have delamination happening here. So I think it's quite a beautiful example of how function and the form of the organ itself feeds back to its own patterning. Continuing on the theme of geometry, we also see, and of course we have kind of put these ideas out recently. So if you are interested in learning more about fractures or about the story, please feel free to kind of check it out. But what we also see, now continuing on the theme of tissue growth and mechanical adaptation. So kind of move to the other part. So as we know that the heart is kind of almost beating all the time. So of course, somehow it has to adopt some sort of mechanism to kind of make it a much more resilient tissue, and it also has to grow. And actually, indeed, we see that between 3 to 5 days post-fertilization, the size of the ventricle almost doubles. So it grows up by almost 90%. So that is interesting. So how do we grow a tissue? By proliferation to increase the cell number. So we quantify cell number. The cell number kind of stays quite similar. So basically, there is not much proliferation happening in this tissue. So that was interesting. So what's the other mechanism? We can think of cell shape changes. And indeed, what we see that as the heart develops, these outer layer of cells, they become much bigger. So they become stretched. And this is a true stretch. We know that the volume remains conserved, and they are actually expanding their apical area. And that's how the heart kind of increases its area. So how does this stretch happen? Because for the cell to undergo that level of deformation, it has to kind of somehow changes its mechanical properties. So we looked at the canonical culprit cell around the ectomycin, cadherin, and all that. And what we do see, here we are looking at phosphomycin, which is a marker for active tension. And we do see that the tension goes down. So in the beginning, you have this cortical phosphomycin, but by the time these cells are stretched, this phosphomycin goes down, and we can also quantify this here. So basically, what is happening is that by lowering their ectomycin tension, these cells are becoming much more deformable, much more malleable, which allows them to undergo such extreme stretching. And then ultimately, you have this tissue growth happening. And we also know this is somehow downstream of Notch. So Notch somehow regulates ectomycin, which leads to this slope here. So that was interesting, but I think again, I think that's something, because every time we look at embryos, there is something puzzling with this. You will get across an observation which you are not able to explain this, and this we also see here. So basically what we wanted to do, we wanted to understand how this ectomycin dynamics influence tissue growth. So we again went back to the theory collaborators, and we asked them that can we develop a vortex model to kind of understand the growth dynamics of this tissue. So here we have developed a 3D vortex model. And what we are doing, we are kind of increasing the frequency of cells with low tension. So these cells are marked in green. And then we plot these cells, frequency of these cells against tissue area. And interestingly, what our model predicted, there is a non-linear relationship between these two parameters. So basically what you see that at the low fraction, you don't see much tissue growth happening. But as soon as these fraction of cells hits a magic number, which is like around 40%, there's a sharp increase in tissue area. And that is interesting. So we thought, is this happening in real also in embryos? Okay. So we kind of went back to embryos, we kind of collected data. And you can see this is model in the solid line, and these are the actual data from the embryos. And you can see that the data kind of follows very well with what our modeling predicted. So basically at the lower fraction, the tissue does not grow, but as soon as the fraction of cells kind of reaches around 40%, the tissue does not care. So the enough number of cells are, softer and just going to expand and increase the surface area. So for people who, I think some of you might have already guessed that this has signatures of critical transition or phase transition like behavior. And I think we have started to appreciate that developing systems, they do utilize these fundamental physical concepts to kind of trigger like morphogenetic changes at much higher scale and much more efficiency. So instead of kind of growing the tissue incrementally, one by one, as soon as it reaches this particular number, the tissue just decides to grow. So that was, again, kind of telling you that how do you, when you integrate different kind of principles and disciplines into your research, it does lead to some sort of observation. So again, we recently published this recently last year. So if you're interested to understand how this cell shape, organ function, they feedback to ultimately make a bigger tissue, please feel free to check this out. I think one thing also I would like to point out, this was, again, this tells us that if in this case, because the tissue has to grow very fast, because the volume of the heart determines how much blood it can pump. But there's no proliferation. So the system has adopted some clever strategies to grow fast without undergoing proliferation, which is a much more intense process. And I think again, this tells you about the wonders of morphogenesis. So that was interesting, but this is something which we have kind of stumbled upon now, and pun intended, but it kind of keeps us puzzled because these cells, they get puzzled as they become stretched. So what we have realized that as the heart grows and as these cells, they become bigger, if you look at their junctions here, you can see in the beginning they have relatively linear junctions, but by that time, they kind of adopt this very interdigitated lobulated morphology, and they acquire these puzzle shapes. So that was interesting. So when we first kind of observed this, I think our hunch was that this is just your excess membrane reservoir. So if the heart stretches in the diastole, these membrane reservoirs or these interdigitations should be resolved. So we don't have beating hearts, but that's not true. So if you look at this particular junction here, if you can follow, you can see that this junction kind of stays puzzled throughout the heartbeat, which we have also kind of tried to kind of make it clear in these panels here. So this tells you that this puzzling, these are stable interdigitations. They are not temporary features of these cells. And this is a permanent deformation that the cell has undergone. So these are stable interdigitations. And now what we are trying to understand why this puzzling happened, what are the physiological benefits, and also how this happens. So the first thing was, okay, let's go back and see when the puzzling starts and what's the, can we kind of really understand this quantitatively. So Molly was working on this project. She kind of developed this pipeline, which I will not explain in detail. But basically, we use solidity as a measurement of puzzliness. So more puzzled, less solid. So she collected lots and lots of data across heart developmental stages. And you can indeed see that in the beginning, you have these linear junctions. But by this time, which is around 15 days post-fertilization, you see these appearance of these very indented, lobulated junctions. And also the solidity goes down, which means cells are becoming much more puzzled. So that's interesting. But then the question was, how cells get puzzled? Why they are getting puzzled? So again, we went back to our theory collaborators and we asked them, what do you think can be happening? So this is again preliminary, but we think that again, this is the role of ectomycin intention here. So if you impose high tension on these simulations, you don't see puzzling behavior. But if you impose low tension, you start seeing these puzzling. And currently, we think that this might be kind of related to the buckling stability. So if you have low tension, you do this, the junctions might buckle. In agreement with this, we do think that these cells have low tension. So here we have jet actin, no, sorry, and alpha 18, which is the canonical marker of ectomyosin tension, and both of these parameters they kind of group down. But I don't think so the situation is as simple as I'm telling you here, but I think there's also a role of more tissue level feedback here. The reason why we see this, because if you look at the diversity of these puzzle cells, so this tells you that the distribution of these puzzle cells in a particular 7B ppf heart. So first of all, they come in all form and shape and flavor. And also their distribution is kind of quite heterogeneous. But anecdotally, what we always see, mostly see, that these puzzle cells are very much focused on these extreme poles of the heart. So here you see puzzling and here you see puzzling. Now, also anecdotally, what we know, we are yet to quantify it properly, that this part of the heart undergoes more isotropic stretch. So this part undergoes more anisotropic stretch, and this part is stretching more isotropically. And that might ultimately contribute to the buzzing behavior of these cells. Again, this is not something unique. In plants, they know that isotropic stretch might lead to these interdigitated junctions. And we also know that this puzzling is indeed affected by the forces. So if you manipulate forces, for example, here we have lowered the forces by treating with this drug called nifedipine, what you see that the puzzling kind of becomes basically, it kind of, the cells become puzzled, so the solidity kind of goes higher. So some more forces are required to changes the morphology of these cells. But then, I think I'll be done earlier, which is good. More time for questions. The question is why puzzle? Why do we create these very interesting lobulated junctions? Well, we think that these interlocked junctions, they kind of reinforce adhesion. So you're kind of creating a much more stronger cell-to-cell adhesion. And that increases the rupture threshold of the cells, as a result of which you have a much more mechanically resilient tissue. Now imagine you can imagine this might be extremely important for a tissue like this, which is undergoing that level of seismic deformation throughout the lifetime of an animal. So this tissue remains single layered throughout the lifetime of this animal, and then it beats 3 times per second. It undergoes all this, mechanical deformation. So it makes sense that the cells have to be really, really strongly adhered to each other. And instead of relying on canonical, adhesion modeling, they just make these interdigitated junctions, and perhaps that makes them much more stronger. So that's our hypothesis, but again, this is not only true to biology. There are various examples. For example, in Japanese carpentry, what they do, they kind of interlock these wooden logs together without any nails or glue to kind of create very mechanically strong, durable structure. And also, interlock pavement systems, they work on the same principle. So wherever you need mechanical resilience and durability, these interlock junctions are important. So that's kind of perhaps what is happening here. We're yet to test this. But again, kind of zooming out, why this is important. Now, most of our understanding of epithelial mechanics, including me, when I was an alpha staff, I was working on this classical epithelial cells and the CA. They're actually, this is one of my wish. They have this very linear junctions and ectomyosin tension and everything. And I think most of our understanding of cell mechanics or epithelial mechanics are shaped by these classical epithelial shape, which have these linear junctions. But we have started to appreciate that not all developing systems, they kind of follow these rules and think of these linear junctions under those cyclic deformation, which happens at millisecond. Will they be able to sustain, those deformations? Perhaps not. And indeed, we have started to appreciate that in systems where there is a need for much more high mechanical resilience, these pulse shapes, they appear. So I've shown you an example of cardiomyocytes, but in plants, they make puzzle cells because they have to sustain the cell shape under high tubular pressure. In lymphatic endothelium, they have puzzle cells. And of course, in fly, they have puzzle cells. So it looks like whenever there's a need to make a much more mechanically resilient tissue, somehow the system, they do not follow this canonical shape, and they adopt this clever strategy to let's puzzle. So instead of like building, kind of utilizing a lot of energy resources, just by modifying their geometries, I think they build a tissue which is like proof. So to sum it up, I think the overall goal of my lab is to understand how this simple structure, which is a single layer of cells in the beginning, transform into this very complex 3D topological meshwork. And we want to understand the underlying mechanism, both cellular and signaling mechanism and morphodynamics across space and time. And in the quest of doing so, we started to appreciate that the system utilizes some non-canonical rules of morphogenesis. I showed you two examples of fractures, so it breaks to build, and also causality. So basically, it does not follow the classical shape. And also, there are other things where we also realize that the system does not follow the rule which we have kind of been learning from the classical epithelial biology. So I think which kind of tells us that perhaps I think there's a need to expand our overview of how we think about morphogenesis in general. So I think with this, again, I would like to thank the team. As I said, they keep the show up and running. I'm just here to kind of represent them. Most of the work I've shown you today comes from Toby, Chris, Molly, and MC. Our funding agencies, our collaborators. And yes, we have open PhDs and postdoc positions. So if any of this excites you, please do come and talk to me. And thank you for your attention.